just in the popular press. I went in for a blood test and I got cloned by mistake, so this guy is a little copy of himself. Again, the big point here is that that would be reproductive cloning, and nobody's talking about doing anything like that. To get into the difference of what we're actually talking about now, sort of moving from cloning, the kind of cloning frogs, to the idea of cloning in human embryonic stem cells, that's another field that's not really that old. I mean, this is kind of amazing to me when I think about this process that since I've graduated from medical school, even just in 1998, we've been able to move into a whole new world where a whole set of topics of science that didn't even exist before 11 years ago is really on the forefront of actually happening right now. Now, how does that happen, and how do I tie together what I was telling you before about embryonic stem cells and in vitro fertilization, and why would the two even be around? So remember, we said Kate and Nate had at least 6 offspring, and they probably actually had other cell masses that they didn't actually end up implanting to try and have babies. They said 6 was enough, right? And if families decide to donate those to science, one of the things that you can do is you can take this little lump of cells that's in the center, that lump of cells right there. If you take those out and you put those cells onto a petri dish and grow them, just this lump of cells, for some specific reasons about the way that they're developing, what they're sort of driving the developmental process, these cells can then keep making copies of themselves, and they're still copies of themselves. So if you think about that article that I mentioned from 1998, the first time anybody tried to make stem cells, those stem cells have gone through this exact same process. They're still around today. They still have the potential to make any tissue in the body. And they're actually cells that are in scientific studies that I'm going to tell you about closer to the end of the talk. Now, in addition to being able to make individual copies of exact copies of themselves and stay around for 10, 11, 20 years, the other thing that they can make is they can make any tissue in the body. So they could make, say, repair tissue that would go into a spinal cord and be able to rebuild the spinal cord and change outcome for patients who've had spinal cord injuries. And that's something that's really pretty exciting and actually really in the process of happening right now.